Screening techniques in industrial microbiology. It is the most important and basic step in industrial microbiology. So, it is the process of detecting and isolating desired microorganisms from natural sources like soil, water sample, etc. In this process, the organism is screened to detect the ability of the organism to produce industrially valuable products. In the screening process, the organism is screened for the production of products like antibiotic, amino acid, vitamins, growth factors, enzyme, organic acid and other industrially important product by the microorganisms. There are two types of screening process employed in industrial microbiology to screen for the microorganism. One is known as primary screening and other one is known as secondary screening. Primary screening is a very easy and quick method to isolate microbes having the industrial potential. Primary screening is simply a qualitative technique in which the organism is just screened to find out whether the organism is producing any desired product or not. Primary screening can be carried out in various ways. Some of the important techniques include crowded plate technique, oxonography, enrichment, culturing. First one is crowded plate technique. It is a technique used to isolate antibiotic producing organism from the soil sample. Second technique is oxonography. It is a technique used to isolate organism producing growth factors and vitamins, etc. Third technique is enrichment technique. It is used to isolate the organism having the desired potential by growing them in specific media or by changing the nutritional composition. Depending upon the type of product, we can use any one of the methods like crowded plate technique, oxonography, enrichment technique. Next type of screening is secondary screening. So after performing the primary screening and selecting the organism, the next step is to find out the true potential of the organism isolated during the primary screening. This is done through a process called as secondary screening. Secondary screening is a type of quantitative based approach. So in secondary screening, parameters like yielding potential, optimal fermentation parameters, whether the product produced is a desired one or not, whether it is a new product or novel product or not, such various parameters can be screened during the secondary screening. Some of the other parameters which can be screened in secondary screening includes product characterization, that is we can just study the type of product produced, its chemical structure, etc. By performing secondary screening, we can also find out the nutritional requirement of the organism isolated during the primary screening. Secondary screening helps in sorting out the organism not having the true potentials like having very low yield, not producing a novel or a new product, having complex media requirement, etc. Some of the common methods of secondary screening include culturing the organisms in liquid media, carrying out the fermentation on small scale to optimize the parameters like pH, temperature, oxygen requirement, etc. Other method is giant colony technique. So this technique is used to detect the antibiotic produced by the organism. So in this technique, the organism isolated by crowded plate technique is further screened to detect the true potential of the antibiotic. In this technique, the organism isolated during the primary screening by crowded plate technique is tested against the test organism. So by testing against the test organism, the true potential of the antibiotic producing organism isolated by crowded plate technique can be verified. By performing the primary screening and secondary screening, the organism having the potential to produce desired product or industrially important products can be screened from the larger population and employed in industrial fermentation process. I hope this video helped you in understanding the screening process in industrial microbiology. For detailed notes on the topics, comment these topics in the comment section. Thank you.